Well, I do have to congratulate all of you this morning for coming. I know it would have been tempting to go. It's too cold out there. I just want to back to bed. This will pass through, I think. Not for what? Not for March. Well, we're approaching March. I thought I'd give myself an opportunity to do a little bit of review on statistics based upon the first quiz you took. First quiz was kind of hard, and particularly because we hadn't covered the material yet. The class mean of the quiz was 7. This is the mean score for each of the lab sections, Monday, Tuesday, Wednesday, Thursday. Which of those lab sections had the most variation among individuals in those sections as far as quiz results? C. Everybody agree? Why? What tells you that it's C? Very high standard deviation. Okay. So there's a big range between the mean and plus or minus 2 standard deviations that would account for 95% of the quiz scores. Okay? Which of the lab sections did best on the quiz? Okay, if we're going to rank them numerically, it would appear that number 1, number 2, number 3, number 4. Okay? But what's another question we have to ask? Can we just look at the means and rank them numerically to determine which one was really doing the best? Well, we have to account for standard deviation, you're right. But we have to determine if any of those lab sections did significantly better than any of the other lab sections. Did they? Here's your data down here that you need to answer that question. What are these values? Those are calculated t-values based upon the class data. And how do we interpret this? If we compare the mean of section A to section B, then this is the calculated t-value for comparing those 2 lab sections and making that determination. Is this a significantly higher mean than this is? As you look at these kind of comparisons, which compare all the possible combinations, are any of them significantly different? Remember back from your lab last semester with the faces and things like that? What's kind of the ballpark t-value that these would have to exceed in order for the differences to be significant? Around 2. Now, does that value change? Yeah, it will change depending upon, remember that t-table? To determine significant differences, you say, is this value greater than the value in the t-table for the appropriate degrees of freedom? Coming back to it all? Okay. What is the degrees of freedom for comparing section A to section B? Remember that degrees of freedom is calculated by this formula, n minus 1 plus n minus 1. You know the n value up there, right? 13 for lab section A, 13 for lab section B. So degrees of freedom is 24. So if you were trying to determine whether this was a significant difference, you would have to go to a t-table, and at the very minimum, you would have to look at that column that said 95% confidence, because we want to make sure we're at least 95% confident the difference is significant. And we would then go to degrees of freedom row 24, and we would see, does this value exceed that value of degrees of freedom 24, 95% confidence? And it's not even going to be close. That value is going to be somewhere around 2. So even though there was a difference numerically in the means, and it appears that particularly that this lab section right here did worse than this lab section, statistically there is no difference. And that is in part because of the high level of standard deviation in those populations. If the standard deviation were lower, then these differences might be significant. but that would be reflected in higher t-values then. Okay, so if standard deviation is lower, then these numerical values that are calculated would have been higher. You're going to have to use this stuff again in lab, okay? Dr. Morton shaking his head back there. So kind of start remembering about how to do the statistics and how to do stat book, because these are going to be tools you will continue to use, not only in this class, but in other classes you'll take as well. As far as quiz is concerned, let me make a recommendation to you. At the end of the chapter, in each chapter, your author has gone to a lot of work to make chapter summaries for you at the end. And if you're going to take a page of notes into that quiz, I would use those chapter summaries as a guideline for, are you getting the proper information written down in your notes? I also kind of scan some of the notes sometimes, and the answers to some of those quiz questions are in your notes. But evidently you're not reading your own notes carefully enough and translating those over into answering quiz questions. We will drop at least one quiz, and so if this first quiz was a little bit of a wake-up call for you, it won't damage your final grade. We may drop even as many as 2, we'll see. Some people did quite well. We had a couple of scores of 11 out of 10. I had 11 questions, but only 10-point base, and so you effectively could get a bonus point if you got them all correct. So some people did it, so just work at it and try to take some better notes, and we'll see what happens next quiz. I'll give you the assignment for next quiz tomorrow. Figure out how far we get today. So any questions? Okay, we're pretty much ready to finish our tour of the cell, Chapter 4. And actually, these last few slides are actually part of Chapter 4, but they very effectively transition over into Chapter 5, which is cell membrane and transport phenomena. So, on Tuesday, at the end of the period, we started talking a little bit about this extracellular matrix, and talked about the fact that on the surface of cell membranes is a fairly complicated group of molecules which have been secreted by the cell. Okay, so think about what that means for a second. If these are molecules that are proteins, then they're going to be made on rough ER, they're going to be packaged up into vesicles, and then they might be directly transported to the cell membrane, where they are then released onto the surface. But very likely, those are going to have been modified a little bit before they're released. So those ER vesicles will likely go to the Golgi apparatus. And in the Golgi apparatus, they can be exposed to enzymes that add sugars to those proteins, making glycoproteins, or maybe add some lipids onto those proteins, making lipoproteins. And then more vesicles pinch off of the Golgi, and eventually they will fuse with the membrane and add to this complex group of molecules. And we talked a little bit about how the fact that these molecules right here make kind of a cell signature for the cell. An identifier, if you like, that among some things say, this cell belongs to this organism. And therefore, immune system, please leave it alone. Only attack things that have a cell signature that suggests foreign. Something that's a potential threat. So some of those immune cells that we talked about, particularly those macrophages, which are able to move through the body by amoeboid motion. Do you remember what part of the cytoskeleton is involved in amoeboid motion?
The actin.
Yeah, the actin filaments. Okay. Then those white blood cells will survey the surface of the cell sand counter. They'll check out these molecules and see, does that cell signature match? And if it does, they'll leave those cells alone. But if it doesn't, then a series of events is going to take place, including likely that cell might be engulfed by that macrophage. And that is the beginning of the process of trying to eliminate that particular threat. We also talked about the fact that the extracellular fluid in all of these molecules out here provides a mechanism whereby things that happen out here in the environment can actually change events that are occurring in the cytoplasm of the cell, including expression of genes. So molecules out here, or stimuli out here, can actually result in new genes being transcribed in that cell. So there's a very important communication factor. And we mentioned the role, and this is one of the quiz questions, that these integrins can play in kind of translating that stimulus out here into action inside of here.
Why did you say it changes genes?
We haven't talked about how it does yet,
No.
but why? Well, because there's something going on out here that the cell needs to respond to. And the way the cell responds typically is by making different proteins. Okay? Plant cells have a cellular structure called a plasmodesmata that allows one plant cell to communicate with another plant cell chemically. These are actually cytoplasmic bridges that join the cytoplasm of this cell to the cytoplasm of this cell. What's this region right in here between the two cells? What do plant cells have on their external surface? Cell wall. Okay. So this is a cell wall from this plant cell, a cell wall from this plant cell, and that cytoplasmic bridge actually punches through, if you like, and traverses that cell wall. Do plants have a nervous system? Not that we know of. Okay, they don't have neurons, they don't have a brain. Do plants respond to stimuli, to external stimuli? Absolutely. Okay, we saw it in the fall, for example. In response to shortening day length and cooling temperatures, plants drop their leaves, for example. What would happen to the growth of a plant if I set a potted plant over there in that window with unidirectional light coming at it from just the outside? What would be its growth response?
It would move towards the light.
It bends towards the light. And many people will incorrectly say, well, that plant is trying to get to the light because it needs more light. Well, that implies conscious thought. Plants can't think. But they are responding. But they don't have a nervous system. What's another way that we can respond to external changes in our environment besides a nervous system? We've got another system. What is that? Pardon me?
I was going to say, like, our body tries to maintain homeostasis.
Our body definitely tries to maintain homeostasis. What if you are walking down the street and all of a sudden 5-7 foot guys come walking down the street with mean looks on their face, swinging chains and looking at you going, Are you going to respond? Yeah. What kind of response is that going to be typically? Scared?
The fight or flight
A
response.
fight or flight response. Okay, your body really gets geared up. And what's going to be responsible for kind of initiating that response? Adrenaline, which is what kind of molecule? Well, a hormone. So we have a hormonal system, okay, that allows us to respond to external changes in our environment. Do plants have hormones? Yes, they do. Okay.
Aspirin. It's either aspirin or Tylenol. Comes from a plant hormone.
I know aspirin's derived from a plant
Yeah.
tissue, but I didn't realize.
Yeah, because I just remember.
Okay.
reading about it.
That response to light and the growth toward light is actually the result of the accumulation of a plant hormone, a growth hormone, on the dark side of the stem compared to the light side of the stem. So the dark side of the stem grows faster, the light side grows slower, and we get this unidirectional growth. So it's important that plants be able to exchange these hormone molecules so that many cells are responding as opposed to a single one. Absolutely.
Going back to like animal cell syndrome, so like when a female gets pregnant, when the embryo is still like a zygote or a fetus, then like the woman is sick, is like the body is trying to attack those cells, right?
No, I think the illness is really a result of the change in hormones that occurs following pregnancy. Okay? Your immune system is very good at leaving the developing fetus alone, in part because it's enclosed in the amniotic sac. Antibodies can cross the placenta, but antibodies will recognize that this is at least maternally host. And so the illness, I believe, I'm not a physician, is more due to production of hormones that occur during pregnancy. Here is an epithelial cell. So if this is the lumen or the cavity of the intestine, and then down here would be the circulatory system, these epithelial cells show 3 different examples of types of intracellular junctions that are really important and something we need to talk about a little bit in terms of the relationship of one cell to another. The 3 types of junctions that we're going to talk about are the gap junctions, the desmosomes, and the tight junctions. What a tight junction really does is it basically blocks materials from moving in between adjacent cells without at least having to cross a cell membrane. So it basically is a barrier within the membrane. So things will really have to go through that cell membrane and cross its cytoplasm before they can get into the circulatory system. Here you can see a tight junction right here between these individual epithelial cells. We don't want molecules sneaking in between cells because they would then be filtered by the membrane. So these tight junctions really prevent things from moving between cells and force them to move across cell membranes where then selectivity can be exercised. We're going to talk about tight junctions in another context, probably tomorrow, in the context of the cell membrane and preventing protein molecules from moving from one side of the membrane to the other side of the membrane. But we'll come back and talk about that later on. Gap junctions are very much like plasmodesmata. They are little tubules that connect the cytoplasm of one cell to the cytoplasm of another cell. One of the places where there are a significant number of gap junctions that connect cells within a tissue is in cardiac tissue. I don't know how much physiology you've had, but in cardiac tissue, there is an initial excitation of those particular cells that then causes those cells to release ions, which then cause subsequent cells to contract. And so we get this wave of contraction moving across the heart as ions move from cell to cell to cell. They're moving through these gap junctions. So the ability to communicate chemically is made possible by these direct joining bridges from one cell to another. Desmosomes are kind of anchors. This cell produces a set of desmosomes, which are proteins that ultimately are anchored into the cell's cytoskeleton. This cell has a desmosome as well, and it's anchored into its cytoskeleton. Those 2 desmosomes can then interact with one another to form what's almost like a little weld, which reinforces this cell. So tissue that is subject to significant mechanical stress oftentimes has a significant number of desmosomes interacting with other cell desmosomes, so little welds to basically keep them intact. One of the tissues in the human body that has a lot of desmosomes is uterine tissue. Why is it important? Because when childbirth occurs and we start having those violent contractions of the uterine muscle, you don't want that uterine muscle pulling apart. Okay? So this anchoring phenomena can be more important in some cells than the others. So there are several different proteins that are involved in cell membranes that are really important in terms of a cell's interaction with other cells in the body. This was actually the last slide in the previous PowerPoint, and I kind of moved it over to this PowerPoint, just as a good transition. What are these? Little yellow guys right here? Those are bacteria. Okay. And since bacteria are not part of the body's normal, particularly, well, some areas the body has extensive number of bacteria. But let's say this is in the bloodstream. Should you normally have bacteria in your bloodstream? Do you know what you call it when you have bacterial infection in your bloodstream? Sepsis or septicemia. And that's very, very serious and dangerous. It can be life-threatening. What's this guy? It's a white blood cell, and specifically it's a macrophage. And what's it doing? What's it doing to those bacteria? It's about to devour it, but before it devours it, it's checking it out. And it's using these cytoplasmic extensions and molecules on the surface of those membranes to check out the surface of the bacterial cells. And if they don't match up, if this suggests foreign, then one of the things that typically will happen is this will begin to engulf and devour, phagocytosis, that bacterial cell. And that's typically one of the initial events in that process. So some interesting things have to be happening to the membrane of this phagocytic cell in order for it to be able to extend these long protrusions and then eventually kind of grab and surround and engulf that particular threatened organism. So one of the things that this does suggest immediately is the cell membrane is a very dynamic structure. Okay. It's not just like a piece of cellophane. Well, when it comes right down to it, it's pretty fundamental. Without membranes, there wouldn't be cells. And without cells, there's not life. So what makes these little membranes so crucial and so important? Well, at a very simplistic level, the membrane really defines the boundaries of the cell. Anything that's inside of the cell is part of that cell. Anything external to the membrane is considered to be environment. Also, pretty simplistically, the cell membrane holds the cytoplasm together. It contains the cytoplasm. We don't want it all leaking out. The membrane forms a selectively permeable barrier. And that barrier helps to regulate what can enter and leave the cell. They used to use the term for the membrane semi-permeable. I think a better term for membranes are they are differentially permeable. They let some things in, but not other things. They let some things out, but not other things. So that allows the cell to maintain an internal environment inside that might be quite different from what's going on in the outside environment. Well, to understand regulation, we will have to dig a little bit into ion pumps, ion channels. So we'll get into a little bit of detail there later on. As we mentioned earlier on, just a minute ago, that the cell membrane is involved in communication, cell signaling between the external environment and the cytoplasm. And the cell signaling molecules are primarily proteins that are part of the cell membrane. They might be things like those integrin molecules or various receptor molecules. So when we talk about membranes, we're going to have to talk about proteins. And then, proteins in the membrane are also catalysts many times. They're enzymes. And many of these enzymes are also involved in this intracellular signaling. So something in the environment can activate an enzyme in the membrane, and then the membrane can produce a molecule inside the cell, which then has some effect on that cell's function. One of the ones we'll talk a little bit about is this enzyme adenylate cyclase, a cell membrane enzyme that takes ATP and produces this interesting molecule called cyclic AMP. And cyclic AMP is oftentimes referred to as a second messenger. Okay, so let's talk a little bit about membrane permeability and kind of movement of stuff across the membrane, some of the kind of simple stuff. Large and small substances move across the membrane in fundamentally different ways. So small molecules like a glucose molecule, or maybe a calcium ion, or a sodium ion, move through the membrane by actually kind of diffusing or dissolving within the membrane itself. And that particular process is called passive transport. There are two types of passive transport we're going to have to distinguish between, and those are simple and facilitated diffusion. But there's another way that small molecules can move across the membrane, and that's called active transport. Anybody have any idea what the term active is referring to? Okay. It needs energy for it to do that. And what form will the energy likely be? ATP. Okay. Larger molecules, or even cells like a bacterium, are too big to just kind of float across the membrane. And instead, for them to be brought into the cell, the membrane of the cell actually has to pinch in and surround the material, ultimately enclosing it in a membrane-bound vacuole. What do we call that phenomenon where the membrane pinches in and brings large substances into the cell? You told me a couple days ago. That's endocytosis, and a type of endocytosis is this phagocytosis process. There are other types of endocytosis. When large molecules are released from the cell by the vacuoles fusing to the cell membrane and then releasing substances outside, that's endocytosis. Sometimes this is referred to as bulk transport. So we'll talk about the transport of small molecules first, and then we'll end up talking about bulk transport later. Well, a membrane's molecular organization really results in its selective permeability. What is a membrane made up of? What two types of molecules? Phospholipids and proteins. Okay, well, you all know what a protein is, hopefully. It's a polymer of amino acids. And depending upon the order and kind of those amino acids, What level of organization does that refer to? Kind and order of amino acids. Primary structure. Okay? Depending upon the primary structure of the protein, it will then fold up into a characteristic secondary, tertiary, and maybe even quaternary level of organization. The proteins fold. And their shape determines their what? Function. Okay? Shape of structure determines function. Phospholipids. Are modified or substituted triglycerides. Remember what a triglyceride is? It's two components. What are the two components?
Glycerol and fatty acid.
Glycerol and how many fatty acids?
3.
Okay, so if we've got this, it's called this part the glycerol molecule, then there can be 3 fatty acids attached to that. What are the 2 types of fatty acids that those could be? 2 categories. Saturated, unsaturated has to do with double bonds, remember? Which of the fatty acids, instead of being fairly linear, tend to have kinks in them? Saturated or unsaturated? Unsaturated. What causes those kinks to form? Do you remember? Double bonds. Okay, the presence of double bonds between the carbons. The saturated fatty acids, which have single bonds, tend to be more linear. Okay. Well, a phospholipid has 2 fatty acids, and then in place of the 3rd fatty acids is a charged group. It's a complex group of atoms that has a charge. Charge groups are hydrophilic. What does that mean? Hydro means water. What is phil mean? Those are water-loving. They will associate with water. Is sugar hydrophilic or hydrophobic? It is hydrophilic. It will dissolve in water. Hydrophilic things are water soluble. The fatty acids are hydrophobic. They are water hating. They like to get away from water. That's why oils float. Okay, they won't solubilize in water. So this is kind of a schizophrenic molecule, if you like. We represent typically this part of it, the hydrophilic part of it, as a circle. And in the fatty acid chains, the hydrophobic parts as 2 little tails. So this is our typical representation of a phospholipid. Molecules that have both hydrophilic and hydrophobic portions are said to be amphipathic. So what happens to those phospholipids if you throw them in water? What do? I want to dissolve. I want to get away. They can interact with one another. And in cell membranes, where the environment is mostly water, and the cytoplasm has a significant amount of water, how can phospholipids orient themselves so that everybody's happy? They can form a bilayer. Okay? So a double layer of phospholipid molecules, where the hydrophilic portions are associated with the environment and the cytoplasm, and in the core of the phospholipid bilayer are these hydrophobic tails. That's a basic organization of the cell membrane. Now, additionally then, there are a number of protein molecules, some of which span the membrane, Some are on this surface, some are on this surface, and those proteins are going to play a really important part in many of the functions of the membrane. Well, what determines whether a molecule can cross this particular barrier of a membrane? Well, that's determined by some of these characteristics of that molecule. So now we're talking about things that are trying to move across the membrane. What's their size? Are they big or small? What's their chemical composition? Are they themselves polar or nonpolar in nature? And particularly, do they carry a chemical or an electrical charge? So, the easiest one first. Is a membrane more permeable to smaller molecules or larger molecules? Small molecules. Okay, small molecules have a relatively easy time moving across this membrane if the membrane is permeable to that. Larger molecules have a much more difficult time. So size does matter. Are they more permeable to hydrophobic or hydrophilic molecules? As it turns out, they tend to be more permeable to hydrophobic molecules. Because really the issue is getting across this hydrophobic core. And if you are hydrophilic, you're not going to be happy moving into that particular environment. If you're hydrophobic, you can move across this membrane quite easily. So hydrophobic molecules, pretty easy. Membranes are most permeable to things that either don't have a charge or are nonpolar in nature, because nonpolar substances are hydrophobic. Well, how do things get across this membrane? Let's say the membrane is permeable. Let's say you've got something that's small and it's hydrophobic. So it can get across the membrane. But how does it move across the membrane? Why is it going to move from here to here? Well, that's explained by the concept of simple diffusion. And our definition of diffusion will be the following. It is a spontaneous, what does spontaneous mean? Happens on its own. Does it require an input of energy? Nope. Net movement, we'll talk about that in a second, of some substance from an area of its higher concentration to an area of its lower concentration until we achieve an equilibrium. So several things going on here. So, if these squares represent molecules that are small and nonpolar, we have more molecules outside the cell than inside the cell. So a higher concentration. One of the things I want you to be thinking about is that when things are concentrated, they are at a state of high free energy. High energy potential, if you like. These molecules, being less concentrated, represent a lower state of free energy. Now, we haven't defined what free energy is yet. We will later on in the semester. But I want to start throwing that term out because when we talk about cell respiration, for example, free energy changes are going to be really important. So, if the membrane's permeable, what is the natural tendency of molecules going to be? To move from a state of concentration or high free energy to a state of lower free energy. And how long will that continue? Until it's even. Until we've reached an equilibrium. The net movement of molecules will be in this direction. But is it possible that an individual molecule might move in the opposite direction? Yeah. but the net movement will be from high concentration to low concentration. Well, again, we haven't talked about cell energetics, but why are things moving from high free energy to low free energy? Well, we'll see later on that behavior is actually dictated by a very important law of science called the second law of thermodynamics. When we bond atoms together to form molecules, that is also a spontaneous process, and chemical bonding is also explained by the second law of thermodynamics. It's a very interesting and sometimes complex law, but it explains a lot of things that are happening in nature. Chemical bonding and diffusion in this particular case. So, here's some molecules of dye. Here's a membrane that separates pure water. If the membrane is permeable to those particular dye molecules, then as they randomly move and collide with one another, the net movement will tend to be from this side to this side, with occasional molecules moving the other way, until we reach an equilibrium. That's diffusion of a single solute. Diffusion of two solutes. Again, assuming the membrane is permeable to both, the net movement of this molecule dye will be in this direction, but the net movement of this molecule will be in this direction. So each behaves independently of one another. Each is moving in response to its own concentration gradient. So this one's moving from high to low this way, this one's moving from high to low this way. And again, eventually they will establish an equilibrium. So simple diffusion can explain the movement of some very important molecules across cell membranes. Small nonpolar molecules like carbon dioxide and oxygen can move across those membranes strictly in response to concentration differences. So as long as your circulatory system is supplying oxygen to the blood, what will tend to be the net direction that oxygen moves? Will it move from the blood into the cell or from the cell into the blood? Which way is oxygen going to move? If it's high concentration out here, is there anything that will tend to make the concentration of oxygen low in the cell?
Yeah, because they're being used during respiration.
It's being used during cellular respiration, right? So cell respiration will keep the oxygen content low here, and if the blood keeps supplying it here, we'll get a natural net diffusion of oxygen from the blood into the cells to sustain those cells. What about CO2? Which direction is that going to tend to move? Yeah, CO2 accumulates in the cell as a result of cell respiration. Carbon dioxide that moves into the blood is eventually moved to the lungs, where you then exhale that particular gas. So the movement of those two particular gases is governed entirely by diffusion. Yeah, now we get into the interesting one, osmosis. This is a special case of diffusion. And very specifically, when we talk about osmosis, we are talking about only the diffusion of water molecules. And those water molecules are diffusing across a differentially permeable membrane. So oxygen does not move by osmosis. Carbon dioxide doesn't move by osmosis. Calcium ions don't move by osmosis. Only water moves by osmosis. And there's got to be a membrane involved if it's osmosis. Cells and tissues can either gain or lose water by this process of osmosis, depending upon the type of environment that they're in, depending upon what's going on out here. So in some instances, depending upon the environment, water tends to enter the cell. In other instances, depending upon what's going out here, water tends to leave the cell. Ultimately, what the big deal is in terms of direction of where water moves is the solute concentration of the solution. How many things are dissolved in that solution? Okay? We can make up salt solutions, some of which are 10% salt, some are 20%, some are 30%. Okay? If we put tissues or cells in those different solutions, they're going to behave differently depending upon what the solute concentration of that solution is. When you add solute to a solution of pure water, it effectively lowers the concentration of that water. So think of pure water as being 100% water molecules. Think of a 10% salt solution as being 10% salt and 90% water. A 20% solution is 20% salt and 80% water. So the more salt you add, the more the water concentration goes down. What is water moving in response to? Its concentration, water concentration. But because solute affects water concentration, it affects that movement. Well, there's three terms that really describe the tendency of one solution to gain or lose water to another one. And typically when we're talking about solutions, we're talking about the environment and the cytoplasm. So is water going to enter the cytoplasm from the environment, or is it going to leave the cytoplasm and go into the environment? It has to do with solute concentrations. Hypertonic solutions, those that have a lot of solute, very salty solutions, tend to gain water from hypotonic solutions. They have less salt. So if you have less salt, you have more water. If you have more salt, you have less water. So water is going to move from its higher concentration to its lower concentration. From the less salty solutions to the more salty solutions. Isotonic solutions gain and lose water to one another at the same rate. There's an equilibrium. So what will be the solute concentrations of an isotonic solution? They'll be equal. Okay, equal solute concentrations, therefore equal water concentrations. So water will move here, it'll move here, it'll be in equilibrium. So here's a simple experiment you can do. You can take a piece of glass tubing and bend it into a U. And there's a little membrane here that is permeable to water, but is not permeable to the solute that we're going to put in that tube. So the membrane's impermeable to the little green circles, if you will. When we use the term hypertonic and hypotonic, we're using them as comparison. Okay? So is this solution hypertonic or hypotonic compared to this one? Let's call this side A. Is side A hypertonic or hypotonic compared to side B? Hypertonic. Okay, it's more salty. Okay. Where is the water concentration going to be higher? Side B or side A? Less solute molecules, higher the water concentration. Remember, solute lowers water concentration? So if the membrane prevents the solutes from equilibrating, but lets the water through, what's going to happen? The water is going to tend to move from this side to this side. It's going to go from the less salty solution, the hypotonic, to the more salty solution, hypertonic. And this is something we're going to do in lab this week, next week. And one of the things it says at the very beginning to kind of get you to hammer this in your mind is that when you're looking at behavior of cells and whether they gain or lose water, salt sucks. Okay? Salt draws water into it. So this solution is going to tend to gain water, but eventually it's going to have different water levels, and eventually this will reach equilibrium. You may know how? Why will the water not keep flowing this direction, even though this is still hypotonic compared to this?
Because the water concentrations stay the same.
Has to do with another parameter besides concentration can affect movement, and that is pressure. So as this gets higher, there's more pressure on this column, and the pressure pushes the water back this way, even though it continues to flow in response to solute concentration this way. So we can achieve an equilibrium when these two sides achieve a certain balance in terms of height. Okay, we will continue tomorrow. Ran over a minute, sorry. Make sure you sign the sign-up sheet if you didn't do that.